Okay, this is my pleasure to introduce Nathan Clark from the University of Pittsburgh. He and I had dinner last night, of course, as the duly host speaker, and he and I had an interesting conversation because neither one of us could remember when we met. And I don't think it's entirely because of social studies here, although that could be part of it. But more that we're at a very similar career stage, have been for a long time. And, you know, we sort of kept bumping into each other and being like, oh, that's cool stuff. And you have partially overlapping interests. But each time we bumped into each other, we realized there's more overlapping interests, and he's doing still more cool stuff. And so at some point I realized, well, we got to have him out here to give a seminar. We've never collaborated before, but maybe we will. We actually worked on an NSF workshop review report on Convergent and its applications that we're working on, and that's been kind of fun as well. So just in case you have similar kind of random interactions with Nathan Clark, I'll go through kind of the CVs and identify him. He did his bachelor's degree at UT Austin in chemistry, and then a PhD at the other UW, Seattle, with Willie Swanson in genomics, and then a NIH postdoctoral fellowship with Chipper Quadro at Cornell. So he's worked on a lot of different systems, kind of in, you know, evolution, molecular evolution. And then set up his lab at the University of Pittsburgh in 2012, got tenure there, and then decided to give Utah a try for a few years, and then is now back at the University of Pittsburgh, still doing the same sort of work. So I'll take to tell you about his work, but I'll just kind of editorialize a little bit that, you know, one of the things that attracts me to Nathan's style of science is he develops really useful novel computation. But a lot of people do that. He also, I think, often finds the most useful application for it. And I don't mean that in a translational sense. I mean that is developing those tools to answer cool biological questions that you have. And I think that facilitates methods that, you know, work better and are intuitive, get to the answer in ways that are really exciting and interesting. And I'm sure he'll tell us about some of that today. So, Nathan Clark.
Wow, thank you so much for that introduction. It's been really great talking again, and I look forward to actually doing something science together eventually. It's fantastic to be back here in Madison. I've been here a couple times, once as an undergrad. I was telling some people earlier, I came up here in 1997 and went to Stoughton. Did I say it right? The town where they had the cyclotron thing. Anyways, it had something to do with chemistry, but that was a long time ago. But it's great to be back and see a lot of faces I know and meet some new people. In our lab, we're very much interested in evolutionary processes. We take this kind of attitude that nature has already carried out so many millions of experiments for us, and really countless ones. And in some sense, we can just sit back and find new ways to interpret all of the variation that exists already in the natural world. And I know that type of concept is not foreign to this department either. So I love this example of convergent evolution here, where you have two different mammals who decided that gliding from treetop to treetop was a really fun thing to do. Of course, these are completely unrelated to each other. One is a rodent, a sciurid, a true flying squirrel, and this is a marsupial sugar glider, right? And of course, there are other gliding membrane types of mammals, and I have not yet found an excuse to work on these yet. So if you have any ideas, please help me, because I really want to work on these really badly. But they get across kind of one of the core concepts of what we study. And that's getting at kind of adaptive phenotypes in one sense. But I also like to say evolutionary responses as phenotypes, because that includes adaptation, but it also includes changes for other reasons that are triggered by changing selective pressures, because you could also lose traits as in a response to living in a different environment. You know, classically, we've studied genetics is always used in evolution in general, has always looked at different ways of studying adaptive phenotypes to get from genotype to phenotype as well. You know, you can do classic studies where you look at a particular trait, you know, has some kind of beneficial value, and you can do crosses within species. You know, I know like the Pazer lab here would work, perhaps not with these exact mice, but with some others to do that. You can look at regions of the genome that might be positively selected, or look at particular populations and look for specific divergence in a population that experiences different selected pressures than the other populations, right? What I'm going to talk about today is more looking at, why did it do that? That's weird. Phylogenetic timescales instead of within species, looking between species in a paradigm that our little microfield is calling phylo-G2P, or phylogenetic genotype to phenotype mapping. And we can't do classic mapping. We can't cross any of these organisms. They're too far apart. But what we can do is look at their trait divergence across a whole tree of different species and ask what genetic changes they have in common as well when you're looking at a particular trait of interest. So we can look at different patterns of evolution along the tree. We can look at every gene and look at their whole gene evolutionary rate along all the different branches of the tree, and see perhaps if we have this red trait is a convergent trait like the flying squirrels. Perhaps there was an acceleration in the rate of change of a particular gene on those same lineages, and then that is a further clue that this might have something to do with that trait. We could also look at other genetic types of changes like gene loss and duplication, gene family size and contraction, et cetera. And there's a number of different, there are more than this as well, different kind of computational methods that have been introduced to look at these types of patterns and how they correlate. So phylo-G2P is an interesting way to get at adaptive problems. It can also, so it can reveal to us regions of the genome that seem to have responded to any particular selective pressure or have changed when a trait is evolved, and thereby reveal to us new changes that have occurred in those same species. For evolutionary responses we already know and appreciate, it could help us get to the genotype underlying those altered phenotypes, et cetera. And other things I'll be touching on today are some of the computational tools that we've developed to do this. Namely, one of the kind of cutting edge areas that we're pushing forward into is to try to understand regulatory regions in addition to just protein coding genes. Finally, and how all these types of studies can hit on topics that are important for human health as well. So before we embark on the actual biology studies, I need to teach for a little bit. So hopefully we all know, what an evolutionary rate means in general. Here I'm talking about the evolutionary rate of a protein coding gene, for example. How many nucleotide or how many amino acid substitutions are occurring on a particular branch of the phylogenetic tree at any given point in time. And many of us probably already have a good understanding that there are slowly evolving proteins on average, like histones. We have ones that are kind of in between, and then we have things, certain protein coding genes that are very rapidly evolving, like those involved in immune response, where they're quite difficult to align even between rather closely related species. What I want you to appreciate, though, is that when you look in different contexts, let's say in different species, those rates of change themselves are variable, right? And sometimes even more extreme, you might find cases where hemoglobin goes through a burst of change and surpasses the rate of other proteins, et cetera. So their relative rankings versus each other are changing constantly. And this is kind of the fodder that a lot of the methods I'm going to be talking about today are operating on, why these rates are changing, right? Why are they changing? Because they're being acted on by different evolutionary forces. One is adaptive change. Sometimes there is positive selection for particular changes to overcome challenges that can drive faster evolution. We always have to, there's always this force of kind of conservation, of preserving what function is already there in the sequence, which tends to remove proposed changes from populations and leads to a slower rate of evolution. And the amount of conservation on any gene varies actually it's not an all or none phenomenon. It also varies with the degree to which that gene is important for fitness at that point in time, right? There are other factors coming into play, but I think you get the idea that all these different factors reflect something about their environment and about inner workings of the cell and can lead to changes in their rates. Rates vary over time. Thank you. All right. So the first environmental change I'll illustrate this with is with change in response to aquatic lifestyles or in marine mammals. You can imagine at some point in time, there was some kind of like very generic mammal. This is my recreation of it. Probably had fur. And then independently of each other, all these different lineages, came into being. They evolved these kind of highly aquatic, in some cases, 100% dedicated aquatic lifestyles. And just looking at them, you can see some traits they share, reduced appendages, streamlined body plans. Some of them lose hair. Their skin gets really thick. They probably have to perceive their environments differently, et cetera. So we know in studying this type of convergent change, convergent meaning sharing a convergent environment or substrate that they live in, we know about all those types of changes already. However, what our particular goals right now would be trying to identify some of the genetic changes that were underlying those appreciated adaptive traits, and then also trying to reveal some previously unappreciated changes associated with this environment. Right? So how can we do that? Well, there are a lot of different ways. But first, I want to have a little teachable moment again. This kind of goes back to something I was talking to Brett about earlier. When I'm talking about these types of convergent phenomena, it's important to remember that can apply to many different levels, right? These species are convergent really only in the wide sense that they live in water together, right? They eat different things, et cetera, but they live in water, right? So that's a very high level convergence. We could talk about specific traits like I did previously that they share changes in. Does it happen at the organ level? Does it happen at the network level, or are we going to look at specific genetic elements? But what we're going to do is deploy a program to scan their genomes to look at specific genetic elements at which they show similar patterns of evolution and then kind of integrate up from there to see what that teaches us about the higher levels of organization as well, right? I'm not going to talk about convergence at single amino acids. So don't ask me, please. All right. So what would this look like in a kind of illustrated example? Imagine we have a collection of genomes from all of these different species. And I've highlighted in blue the kind of aquatic species in this teaching example. And the branch links here are representing the average evolutionary rates across all protein coding genes for these species. So it's kind of the expectation, if you will. Then we can study individual protein coding genes one by one. Here is a protein involved in signal transduction in the sense of taste. So it's GNAT3. From its alignment, you can actually already see a few changes there, specifically in the dolphin and the orca. That might be better reflected in this tree. You can see those same two species have a really long branch leading to them, meaning they experience more amino acid change than others. And so if you squint your eyes, you might be able to see that the aquatic species here have longer branches, but that's not a good way to do science. So what we instead do is we normalize these branch lengths by the expected branch length, and that gives us kind of a normalized rate of evolution. So we can look and see whether there are proportionally more changes in those species. So here's what it would look like. You take GNAT3, a single protein, you kind of normalize out the expectation. From the residuals there, we get these relative evolutionary rates. And those represent, a positive value is more accelerated evolution, and a negative value is decelerated. And it doesn't take a genius here to see that the red dots here, the aquatic species, have a higher rate of evolution in GNAT3 than all of the terrestrial species, right? And you can do statistics on this and show it with numbers as well, right? So boom, this has a very low p-value, rejecting the fact that rejecting the hypothesis that they're from the same rate class. We can repeat this for all genes in the genome, and you get a table that looks like this. This is the very top if they're ranked by low p-values first, and the list would, of course, extend way down into the center of the Earth. I don't expect you to be able to read this, but there are lots of different functions represented here, lots of different gene families that are rapidly evolving in aquatic marine species. And these come from however, from a kind of a choice set of functions, though. There's a lot of proteins involved in the senses of olfaction, taste, cartilage and connective tissue, hair formation, skin, nervous system, etc. And so it's actually quite a biased set of genes there at the top. We also then went and did tests for positive selection. There are two reasons any gene can be accelerated on a particular branch. It could be because there is a lack of constraint or less constraint, or it could be almost the exact opposite case, that it's evolving adaptively, that it's rapid, right? So we did statistical tests to determine that, and actually just the choice few of them have anything to do with positive selection. Most of them are due to just relaxed constraint in the aquatic area. So remember, we're looking at a much longer version of this list, but you can kind of, oh, and there's a statistical excess of these. When you summarize them looking at Go terms that are significantly overrepresented at like a 5% false discovery rate, here are the kinds of terms you end up with. By and large, it's the senses of taste and smell, lung function, skin and connective tissue, muscle, and a lot of nervous system genes. So these are what we saw accelerating in marine mammals. Some of these make sense, like the senses of taste and smell. Perhaps wasn't surprising. We also noticed, if we look carefully, a lot of the olfactory receptors in these species were pseudogenes and rapidly becoming so if they weren't. And this fits with observational knowledge that dolphins and whales don't smell at all, right? We've known this behaviorally since like the 60s. And if you look at their brain, they just have no olfactory bulb. It's just completely gone. The olfactory bulb is a specialized area in our brains for smelling, of course. Also, it was the same case with taste. Again, it was known to be absent in dolphins and whales, but what we were able to show is that this is a trend that is more general across aquatic mammals as well. Even manatees and dugongs and seals and sea lions are kind of losing their senses of taste and smell. I don't know why, and no one does. Some people propose it might be because they have strong visual cues or echolocation, or they just swallow their food whole anyway. So why would you care to taste? But the jury's still kind of out on that. So by and large, you could say that the sense of taste and smell are accelerated because of loss of function, right? And that's true of this category and as well as these down here. It turns out these nervous system ones were a bit confusing to me, and we proposed in the paper that it might be because they are more specific to the olfactory bulb in the brain. They're nervous system families of genes, but they weren't assigned to any specific brain region or whatever. So I actually went back a few days ago and looked, and sure enough, the top nervous system gene that contributes to this, when you look at single cell data, is in the olfactory bulb. So it was kind of a really nice single data point that seems to corroborate this idea. Now I just need to find time to look at the rest of them. However, the ones that were not showing loss of function are these, the lung and skin functions. They were showing actually the opposite case. Almost all the genes that were accelerated in these showed significant signs of positive selection for amino acid changes. Okay, so they're not losing function. Why? We had some various hypotheses. Perhaps lung and skin need to behave very differently in water. That makes sense. Lungs of aquatic organisms are, every time they dive, they're compressed down severely and then they have to re-expand. Skin, obviously, you're bathed in water constantly. There must be a lot of structural change going on there. That's one hypothesis. The other is pathogens. Whenever you see a positive selection around, that actually needs to be the first hypothesis you come up with. And so I actually, we had a undergrad in the lab, Alex Preble, who's graduating this year. He's undertaken that second hypothesis directly and tried to test more heavily that idea that this increased positive selection is due to a higher pathogen load. It's not a crazy hypothesis because if you look at ocean or lake water under a microscope, it's chock full of microorganisms. You know, relatively, we're sitting here in this air, which is pretty much sterile. And so he's looked at collagens and keratin and skin cell junction proteins and sees indeed that they're the ones that are undergoing higher rates of positive selection, but only the extracellular portions, which would be the portions that bacteria and viruses would try to exploit in the first place, et cetera. And he saw another way he tested the hypothesis is that if the acceleration of change had to do with restructuring of skin to become watertight, then you would expect a burst of adaptive evolution in the beginning, and then it would go back down and everything would become normal again, right? Because dolphins and whales, their last ancestor went into the water about 60 million years ago, right? So they've had time. Well, it turns out the adaptive evolution has continued even in pretty much till the present day, right? So it's ongoing, and that's exactly what you would expect if it's the pathogen load that is driving that acceleration. So Alex is now writing that up. I just want to point out quickly that these methods, this study was done using our package, RER Converge, and so it's been developed by all these wonderful people shown here. And it was recently used in a study of extreme lifespan and rockfish from Peter Sudman's group at Berkeley, and which got the cover art that month. So it's really pretty awesome. Okay, so there's another aquatic story I like to go into, and I think all the students who had to read our papers, this is the one. Okay, so we also thought it would be fun, instead of just looking at rates of evolution, to just ask a more simple question, really. What are the genes that become inactivated specifically in aquatic mammals? Right. So we again started with many terrestrial and aquatic mammals all grouped together. We had actually a few more than this, but this is just so I can show it on a slide. And for all of these genomes, we went through and looked for obvious signs of pseudogenization, like a genetic lesion, a frame shift, a big deletion, a stop codon where it shouldn't be. And so we just scored every gene as 1 or 0, you know, functional or pseudogene in all these different species across the board, and use that as raw data to go through and ask which genes show preferential deactivation in the aquatic species at the same time preferential functional status in the terrestrials. When we did this in a phylogenetic framework called Bayes traits, here's the top 10 genes from that. Of course, it goes much further down. Hey, we already see the olfactory receptors and taste receptors up at the top again. That's exactly what we would have expected. And so that was kind of a nice positive control that this worked. We've already talked about this. However, there are a couple genes mixed in here that have nothing to do with taste and smell. One is an antioxidant, glutathione S-transferase. And the very top gene, like the most extreme one that was never lost in a terrestrial species and had a really high apparent rate of loss in aquatics, was paraoxinase 1, which I almost fell out of my chair when Claire, an undergraduate at the time of the lab, came and showed me this list. She and I kind of programmed this together. And it happened that where I studied my grad program at UW in Seattle, there was one researcher I knew in the department who only studied that gene. So I knew exactly what this gene was. And I was like, what is going on? Because all I knew is that PON1 was there because it breaks down organophosphate pesticides, right? This is why Clem studied it. So what in the world is that doing there? I just want to show a little more details here because sometimes the interesting parts from the details. Here's PON1 and its various genetic lesions and some of the original species we looked at. You can see that all the cetaceans share, every cetacean shares these exact same 6 genetic lesions, which is very clear evidence that it was already inactivated in their last common ancestor. This is also true of the sirenians, manatees and dugongs, and the extinct stellar sea cow. They all have this early stop codon. So again, it was deactivated very early. Pinnipeds were more of a mixed bag. They're much more recent aquatic mammals. Some showed genetic lesions, some didn't show obvious ones, but I'll come back to that. We've expanded this, many-fold greater than this. We've now examined something more than 400 species. Every terrestrial species has a functional PON1. And now we've shown pretty much every aquatic species has a non-functional PON1, right? We've looked in beavers and otters, and I'll get to the pinnipeds in a second, and they're just all losing it. Capybaras still have a functional one, maybe because they're kind of new to the water. I don't know why. But... Okay, so part of the other idea is we were just looking at things that might come much later, stop codons and frame shifts. What if we just look at expression? Luckily, PON1 we know is only expressed in the liver because it's a bloodstream enzyme. So it's really easy to get liver from species, actually. So Allie Graham, who just got her own independent position in Kansas, went through and got liver samples in any way she could. And we went through museums and, you name it. And we got liver from a bunch of different species. And what she find, of course, corroborates in cetaceans, you see no expression of PON1, whereas in related ungulates, you see good levels. In pinnipeds, you see a highly depressed expression of PON1 and much less than any carnivores that are around, essentially. Interestingly, in beavers, she also found no expression. So it very matches what we see. But what it shows is a lot of these pinnipeds that have zero expression at the mRNA level were the ones that weren't showing genetic lesions early on, right? So it seems like the first step in this phenomenon is that the gene is downregulated very rapidly for whatever reason. When we look at PON2, which is not very functionally related to PON1, it's not in the bloodstream, it's intracellular. So this kind of serves as a control. It has different substrates. It had more normal levels. There is this interesting, sorry, I didn't point out for PON1, this interesting pinniped that has just completely unfazed. I don't know why. So we need to study this one. And there it is again. What's going on with that guy? So PON3, we know much less about PON3, but interestingly, it shows a very similar pattern. No expression in beaver, no expression in pinnipeds except for that one outlier. And interestingly, not depressed in the cetaceans. Don't know why. But in general, it's much more lowly expressed. Okay. I'll speed things up a little bit, but let's just say I ended up going back to work with Clem for long at UW, and Rebecca and Judith, who were also there, where we just biochemically measured PON1 biochemical activity and blood plasma from all these different species, and it corroborates everything. You see no activity in cetaceans, depressed activity in all the pinnipeds, or almost none, basically baseline, and same thing with the beavers. So it becomes even more apparent when you look at the blood plasma. So the model for this is the following. It seems like, we terrestrials, we have a lot of PON1 protein doing what it should. Gene transcription's happening, it makes functional protein, we're good. But for some reason, when species start becoming aquatic divers and such, the first thing they seem to do is reduce the PON1 protein levels that they're producing. Then other subsequent genetic lesions and downregulation occurs, produces less protein. What's there is probably less functional until eventually you get a deactivation and you get a complete pseudogene and dead enzyme. And that's where the sirenians and cetaceans and some of the pinnipeds are sitting. Right? So you've been very patient. I've neglected to tell you what PON1 does in the first place. And that's kind of partially on purpose. Biochemists think we have a good idea of what it does. I don't think we do. We know, here's what we know. PON1 alleles come up every time we do a genome-wide association study for heart disease, atherosclerosis. So it has something to do with plaque formation, perhaps, right? Biochemists looking for its natural substrates have found that it can break down oxidized fatty acids. So except for this red part, this would be a healthy fatty acid. When they get oxidized, then they become sticky and bad and can form plaques. So therefore, pond 1 breaks these down so that they can become excreted and broken down in other ways. Okay? And perhaps that is what part of pond 1's function. Pond 1 actually got its name early on because it was found to break down organophosphate compounds, which are nerve agents in chemical warfare and also pesticides. Clearly, that's not what pond 1 evolved to do because we've only been making those for less than 100 years. But it just serendipitously has that function. And that's why when we spray it on our agricultural fields, we don't drop dead, but the insects do, right? Because they don't have pond 1. One of the consequences of that, however, that we pointed out is we have these innocent little, well, large creatures like manatees that are living up in the freshwater systems in Florida, shown here in red. These are or orange. These are all protected zones for manatees. They go up inland quite a bit. And they're absolutely adjacent to all this agricultural land shown in green, such that the runoff from them is dumping directly into their waterways. So here we have people using chlorpyrifos, which is one of these pesticides that could then be getting into the waterways where these poor manatees are sitting that literally have no enzymatic defense against these pesticides whatsoever. And this is the only defense we have against these organophosphate nerve agents. So was that leading to their decline? Probably not, but it might have been contributing in the sense that it made them more lethargic. They're getting hit by boats, who knows? But luckily, since this paper came out, chlorpyrifos was banned. It was reversed in the first Trump administration, but maybe it got banned again. I don't know where it sits right now. But. And this led to kind of a nice flurry of news articles where we got to talk with heroes like Ed Yong and other, Carl Zimmer, big writers that I've always read, and got to actually meet them. So that was fun. Okay, so why in the world is this thing lost? I've also neglected to get to that. And the bottom line is we don't know. It seems kind of strange and paradoxical, actually. The baseline we should probably consider is kind of a more neutral case. The null case is that it's no longer important in these species for some reason, and it gets lost. However, we feel like there are some certain arrows pointing to an adaptive loss because it is the absolutely most consistently lost thing in aquatic mammals, period, right? More than all the hundreds of other olfactory receptors that exist in their genomes. This one gets lesions right away, very fast. It's also lost very quickly. We can tell it was lost in the last common ancestor of all these things, not later on in evolution. Eventually, it was lost right away. And we also go on to believe, we hypothesize it's related to diving. And that's because, because diving leads to ischemia in most of the body, right? These prolonged dives, you get really depressed or zero oxygen at the end of one of these dives. Some of these creatures stay underwater for like an hour, right? And deplete all the oxygen except to their brain and heart. When that happens in our tissues, we get an inflammatory response, right? Massive, right? And it leads, in one of the parts of inflammation is producing reactive oxygen species, which oxidize the crap out of everything, right? Excuse me. But so that is a huge stress on these species. And like I stated before, here's the paradox. We think that pond 1 is there to dispose of oxidatively damaged lipids. So why would you get rid of an enzyme that's cleaning up oxidative damage when you were undergoing a lot of oxidative stress in the first place? It doesn't make any sense. So I think we really need to go back to the starting page here. I'm not going to drag you through this. Basically, its loss kind of correlates with diving ability. Oops. And I think we have a lot left to learn on these. Some people who study diving organisms have found that other oxidative stress defenses have been actually upregulated. Classic enzymes like catalase and superoxide dismutase have constitutively higher levels in the tissues of species like elephant seals, which also dive for very long periods of time, such that, why wouldn't actually pond 1's levels also be upregulated? And my new hypothesis is that actually pond 1 is not there to mop up or after oxidative damage. It's actually there to trigger the inflammation in the first place. And this is somewhat informed by one paper where a biochemist found that PON1 can actually produce certain forms of lipids called eicosanoids. And eicosanoids control inflammation, right? So if PON1 is producing eicosanoids that are promoting this localized inflammation after diving, what's one good way to protect yourself from it is to stop that inflammation right out. So if you remove PON1 and you break that signaling through the eicosanoids, you've solved that problem already. So that's look over the next 5 years or so, hopefully we'll get closer to figuring that out, but that's what we're thinking. I want to show you a few other kind of applications. What time do I need to end by? So I'm going to look at subterranean evolution okay, thanks. And I should pick up pace in general. This is a fun one because these tunneling species, kind of like the aquatic ones, are not related to each other, but they've evolved similar habits and morphologies. Again, through the RER converged kind of pipeline, you can see certain proteins have accelerated, like this lens protein, lens intrinsic membrane protein 2. Some proteins that accelerated were ones like keratin 9 that is expressed in skin, but keratin 9 is only expressed in foot pads in our hands and feet. It's kind of interesting. In fact, it was very accelerated and actually due to complete loss of function, pseudogenization in all these different mole species. And the interesting thing is that when you lose keratin 9, like some humans carrying 1 or 2 defective copies of keratin 9, what happens is this. Isn't that crazy? You get thicker palms of your hands and your feet, right? Which for them is an adaptation. For us, it's just like kind of like a mild genetic disease, right? So I thought that was interesting. Maybe it's this thickening is helping them tunnel better. But by and large, in these subterranean species, what you see is an acceleration of eye-related genes, specifically all the ocular genes related in the retina and the lens. And this is, again, due to loss of function, kind of like the olfactory genes in the aquatic species. It turns out this is a good way to predict new genes. In fact, Courtney in the lab has used this because when you rank genes by their acceleration in moles, you get like lots of the well-known ocular genes already, but interleaved in between them, you have these genes that we know nothing about. And there's not a single paper or database thing. It's just has some name. Or we have so she's picked out these 5 genes that we're going to characterize now by knocking them out to see whether they do affect expression, et cetera. And we're going to do that in zebrafish, actually, because they have them too. Another fun trick is you can kind of do this study backwards. You can start with a set of genes you know are ocular, and then you can use their average rates in all these species to rank the species by how fast they're evolving in those, if that makes sense. So here, as you would expect, the mole-like species have very rapid rates of evolution for the ocular genes, these 189 ocular genes, but so do other species like armadillos and bats and shrews. And why would that be? Well, it turns out armadillos have famously bad eyesight. They have tiny eyes and they really only come out at night. And if you startle them, they'll run into a tree as they're trying to run away from you. They're clearly echolocating bats don't use their eyes much. And these certain types of shrews, for example, have very poor eyesight. They let hair grow in front of their eyes. They just don't care. And so it's neat to see them popping up. So this is kind of another paradigm we can use to learn more about species as well. And I was actually thinking about doing this for some yeast species, as we talked about at dinner with Chris. Whereas well-sighted bats, the fruit bats, have more of an average value. I don't have time to talk about the marsupial mole, which breaks my heart. But basically, Sarah found that they lost their eyesight progressively over more than 10 million years. First, the lens proteins went, then the cone photoreceptors, which are involved in bright light vision, followed by finally the rods gave out later on, and they're still going out, which is kind of interesting. So you can almost imagine this beautiful blonde marsupial mole becoming more and more kind of subterranean, dedicated to its burrowing lifestyle over a period of more than 10 million years. It's kind of fun. Sarah's now replicated this for lots of different mammals, and you get, interestingly, the same progression. Just to sum it up, the lens goes first, followed by cones, and then rods, in most cases. So, okay. We also took a stab at regulatory elements, putative enhancers. For example, around this classic eye development gene, PAX6, a student, Raghav, picked out 150 highly conserved, so this is the phylo-P track, highly conserved genetic elements of unknown function. He combed the literature, and there were 3 known enhancers, ocular enhancers of PAX6, shown with these 3 stars. And when he applied RER Converge to them, those came back as exactly the top 3 scoring enhancers in this study. So kind of giving us promise that we could use the same types of strategies to identify regulatory elements and hook them up to higher level phenotypes, right? So now we're still kind of honing these methods with different approaches. I've talked to some of you about this already today to try to deploy it more genome-wide and to be continued as we improve them. But already we're taking a closer look at certain ones of them that rank highly in the list in zebrafish, where we can take the element and introduce it. We can take the element from zebrafish, that is, and look at reporter gene assays. And in this case, this particular element, 6078, is lighting up in the optic tectum of the brain, which is where it processes visual signals. As well, interestingly, though, as well in these other sensory centers, like the olfactory bulbs and the kind of midline sensing organ back here in the brain as well. More to come on these as well. But finally, we also thought it would be fun, since we started working on moles early on, we teamed up with Children's Hospital clinician, Dr. Ken Nischel. Ken is known for providing genetic diagnoses of children who have ocular abnormalities. And they achieve a genetic diagnosis in roughly 50% of cases, right? So the other 50% he sends to us, and we talk about phenotypes, and we do whole genome sequencing on them and their parents, and we try to find the causal mutations. We're just now starting to crank a lot of these through, but in the first 2, Sarah Lucas in the lab found pretty much smoking guns for both of them. They were both de novo mutations, apparently acting dominant in these children, one in a lens protein GGA8 and another in another lens protein crystalline beta. So it's been kind of a fun thing. And our hopes are hopefully eventually we might be able to use even some of this evolutionary information, some of these new regions to kind of fold in here, but we're happy to find them wherever they lie. So what can we talk about, summarize about convergent changes in general up to date? I've took you through a couple of these stories, aquatic and subterranean. We've also looked at hairless mammals, long, lifespan as a more continuous trait, and high altitude in different stories. And what we can see is that there are a number of different categories of genes that appear. In almost all of these, it's actually pretty easy to find genes and things that are discarded, right? They're much easier to detect. Once you lose something, its rate of evolution stays high forever. So it's like it's screaming at you, basically. And so these methods are really good at finding losses of function. That's one thing. In some cases, we were able to identify some things that appear to be more due to new selective pressures and continued function and positive selection, like the skin and cell junction proteins in the aquatic mammals. And one story I didn't go through today, when we looked at lifespan of the trait, which varies from like 100 years to 2 years among mammals, we saw increased, so slower rates of evolution in long-lived species in these specific processes: cell cycle control, DNA repair, telomerase. What do those all share? Any ideas? Well, those are all related to cancer, right? And prevention of cancer. If you're living a long time, you have a large body anyways, you need to be able to prevent those cells from becoming cancerous. So. Okay, so is evolution predictable? I mean, I think we and many people in biology continue working on this problem. I think to some degree, you can make some predictions about what will happen, given what we saw like in aquatic mammals. What if we looked at other aquatic species, you know, like the platypus, we didn't include it yet, or penguins, or sea turtles, or sea snakes. You know, perhaps we would conclude they're very likely to have lost their senses of taste and smell. Maybe their PON1 gene, right? They're all gone. And what about even like ancient extinct things like this mosasaur that I'm very glad is now extinct. Could you imagine if this was like in the lake right over there? Like, I bet it lost its senses of taste and smell. I'm just guessing, but that's my guess. Okay. Oh. All right, I've got a few more minutes. I want to take you through a variant of this where we're looking at the rates of evolution of genes, but rather than correlating genes with phenotypes or environments, what if we looked and tried to correlate genes with other genes? Who's correlated with each other? So you might remember briefly from the mole story, this lens protein showed rapid rates of evolution in the moles and bats and stuff. But so does this other lens protein. That makes sense. They're both in the lens, right? Such that if you plot one versus the other, there's actually a correlation here. And that's kind of cool because what if we didn't know what this gene did? We could automatically, from this correlation, conclude that it probably has something to do with this function. They're co-functional, so it's probably also in the lens. The cute little Cape Golden mole. Right? So we've called this phenomenon evolutionary rate covariation, or ERC, which I later learned is a funding agency in Europe, but too bad, I already have the name. And we can just simply measure it as a correlation coefficient, right? One being a high positive, the highest possible positive correlation, right? So covariation equals cofunction. We can do this, if you look at just two random genes, you usually see something like this, no correlation. If you look at functionally related genes, you see a positive correlation. These are two complement cascade proteins in mammals. And so we can infer, we don't even need to know what's driving the rate changes here, right? In the eye genes we knew, you know, blind, not blind. Here, I don't know what makes complement cascades go faster or slower, and I don't have to know, right? I can just look at their rates. Who correlates? And so we've repeated this kind of study genome-wide between all possible pairs of protein-coding genes in many different organismal groups now, and use that as kind of a fun way to make new inferences. For one, this phenomenon occurs not just at single pairs, but if you look at the entire complement cascade pathway, that was what these proteins were doing. There are many high and positive values if you look at all pairs of complement cascades, such that their mean value shown out here at about 0.3 something, you would never get a value that high if you just randomly chose sets of proteins from the genome, right? So in general, most protein complexes and pathways are significantly correlated in this kind of way when you look across these species. And so what we've been doing is teaming up with groups, experimental groups all over. This is with Adam Kwiatkowski's group, who studies E. cadherin and in Drosophila. Now let's just say they knew some genes over here that participate in E. cadherin functions. And we just fed him this list in the middle of genes that are highly correlated with these genes. And then they subjected them to this assay where basically all you need to know is if there's some red, it's positive. It means it's in the pathway or it has the function that they expected. And so you can see roughly half of these genes that we gave them have the positive phenotype, right? Whereas the other half didn't. We even convinced them to do some random negative controls from the genome. It was a very laborious test. So they did 6. That was all I could get them to do. But anyways, none of those showed the positive phenotype. So we've done this with many different researchers now, basically trying to help them find new genes in their function of interest. And that's reflected in all these different publications and all these different systems here. And it's been written up as this kind of, in this perspective a little bit ago. So now, the fun part that I should have led with, I've been working with your famous massive yeast collection here as well, using the same technique. And the neat thing is, all these were done on few species, like 18 species, right? So now we're like drinking from the fire hose of species, and this isn't even the biggest one yet. You've got thousands now. But it's already, we had to, and you have lots of mammals too. We had to rewrite, give me more. We had to rewrite all our programs to handle this many species, but it was well worth it because before, with 18 species, we were getting curves of power that look like this. The new methodology itself on that same 18 species already improved things dramatically. But then when we kick it up to 300 species, you get this huge increase in power. This is a very steep curve that's showing statistical power. So it's very much worth it. So we're pretty excited. Jordan, this is Jordan. She's going to graduate this year with her PhD. She's been using it to make like networks of related proteins, and they fall into these very clear, like, hey, look, galactose right there. They all kind of formed a natural network. You get other very highly, I mean, you can draw more stringent thresholds and make smaller subnetworks, but basically you get things that group highly by functions, which is really fun to play with. She did this en masse, starting with protein complexes as well. I only have brief time to show you a few, but basically you can get interactions across different parts of the cell, though. It's kind of fun. So in this edge, zooming into the ATP synthesis area, let's just zoom way into here. Let's look at this little cluster here. Here you have these kind of different mitochondrial complexes involved in the production of ATP. And then way over on the peroxisome, in a whole different organelle, there's this ABC transporter that brings in the ATP. And it's highly correlated with those that are producing the ATP over here in the mitochondrion, right? And so we see a good number of things like this that are confirmatory of what we know about cell biology, and that's really fun. And let's just say we're starting to deploy this with some yeast biologists at Pitt, but hopefully also here eventually to discover new long-range interactions and things going on in different complexes and cell systems. Here's just another example, but for brevity, I shouldn't take you through it. But basically, you see strong correlations from beta fatty acid oxidation to the TCA cycle. This is again between the peroxisomes and mitochondria. And here's one small example where Jordan has pulled up a new protein. She looked at, basically, she said, like, I want to find proteins that are correlated with the nucleus that aren't themselves in the nucleus, right? Or the nuclear membrane. So she was looking, she's picked out, she likes this protein USO1. USO1, and by all reports, is in the Golgi, but it's extremely highly correlated with these nuclear pore proteins for some reason. And we do not know why. But when we dig a little bit, some real, not ancient papers, I don't want to date myself, but some older papers, showed someone did mutate this gene and saw that the nuclear morphology was drastically changed in this, and no one has followed this up since. So maybe there's some reality to this interaction, more to be determined soon. Okay, and we're looking in mammals too, looking for new fertilization genes, et cetera, and knocking them out with some collaborators at USC. So the basic idea, I can wrap up, is genomics has given us kind of this wealth of functional entities in the genome, only some of which we have assigned to higher level functions. Some we have some idea of molecular phenotypes or things, but the idea is we can take these protein coding genes, these millions of completely ununderstood regulatory sequences in RNA genes, and we can kind of start to use some of these methods to link them up into higher level functions, higher level processes to which they contribute. So we can look at lifespan and eyesight, diving, hair, blood feeding and insects, et cetera, whatever we can imagine. And yeah, so I have a lot of people to thank. Here's my current group and some people not. Hey, here's Jared. Everyone say hi to Jared. You remember him, okay? and to alumni who came through. And I must say a lot of the computational work, I've been in a very long collaboration with Professor Maria Chiquina did as well. So a lot of credit goes to her too. Thank you, guys. Yeah.
More effective than black and white ones.
Yes. So in the marine case, those are interesting examples because it was actually a case where the adaptive evolution is ongoing, right? Because the selective pressure, these pathogens, is what we're thinking, are remaining a constant, arms race, right? And so even in that case, we've identified a persistent positive selection event repeatedly going on. And so in that case, we haven't even found for that one, that's not even an example of an episodic one. Those are much harder to find. And we're working right now with Sergey Pond, who has written a lot of these DNDS kinds of methods, hi-fi, busted, all these things, to create a specific set of likelihood models to find such episodic episodes, if that makes sense. So, yeah. So we would like to find them because they're so interesting, but it's harder. Yeah. Yes. Yeah. Yeah. Go ahead. I don't have an answer for that yet. So the PON1 gene its regulatory regions are not characterized. We know where the promoter is because it is transcribed. As far as which enhancers are acting on that, we're trying to identify them. Wynn Meyer, a former postdoc in the lab, I'm letting her do that. And she now has her own group at Lehigh. So she's trying to find where its enhancers are. We just don't know yet. Yeah, there's not even any conserved regions nearby. That's the thing. So yeah, we would like to know. We don't know yet. So we need some open chromatin data or something from liver that would help. Yeah. Yeah.
I never said the word homogenization. I'm interested in that process.
Yeah.
So, you got up to sort of stepwise model, for one example. But in your probing for, different forms of good optimization, do you find that any particular mechanisms for loss are disproportionately represented? I can think about something like, an early stop code on a certain number of
Yeah.
possible places that could happen. So are there any of the individual types of optimization that are disproportionate given the number of possible occurrences?
That's a great idea to look at. I mean, we would want to normalize it by opportunities and we haven't done that. But I think more generally, we could say that the regulatory ones, well, the decrease in expression seems to be the first thing because but what am I trying to say? There is no case where there is a stop codon where it is still being expressed, right? Whereas there are cases where it is not being expressed in which we don't see any obvious genetic lesions, right? So perhaps the regulatory downregulation is the first thing to happen, but that's all I can say on versus which kind of pseudogenization, I don't know. Yeah, not yet. Yeah. Yeah, Fred.
So much power.
Like in cases where there's also relevant phenotypic variation or oh yeah. Oh, clearly. Yeah. Yeah, that would be that could become its own thing or a layer with it to look at you know, putatively deactivating, or just levels of polymorphism is a rough way, right? Or, yeah. Yeah, yeah, yeah. Yeah, and that's a great thing to think about. You know, there are more and more opportunities for that as we sequence more species. Right? Yeah, good point. Yes. Yeah, for the like you know, the tetrapod emergence from the ocean, it's probably too long ago for, I mean, or I just haven't thought deeply enough about it yet. And it essentially, it happened one time, right? So we would lose a lot of power. I mean, I guess it happened again for insects and arthropods and different things, but we do have one publication where a renal scientist, he studies kidneys and stuff, and he brought us along for the ride essentially, where he was studying that, actually, the emergence, and basically there was a huge difference in how they had to deal with salt, right? Salt balances, and he found that these particular regulatory regions that kidney doctors pay a lot of attention to on this ENaC complex for sodium uh, evolved when after we came out of the oceans, which is cool. They don't exist in those regulatory regions on the protein allosterically, don't exist in the aquatic vertebrates. So we found they were coincidental with that emergence, but that was kind of fun. But that's all I know about that. Sorry. Yeah. Yeah. Yeah, I haven't seen telomerase pop up in any other of the various phenotypes we've looked at. Just this body size, longevity, lifespan one. More specifically, you know, its specific effect, I can't tell. And maybe I can elaborate on that a little more. In that whole study, we're not sure whether we can't, using just these methods alone, distinguish whether these changes in selective pressure led to a longer lifespan, or they're just a consequence of that, right? You know what I mean? I suspect maybe more of them are a consequence of having a long lifespan versus enabling it, or if you get what I'm saying. So I think there's a lot of potential there too, but yeah, I was speaking with Audrey earlier about getting back into the lifespan game, because there's a lot we can learn there. So I'm pretty excited this visit maybe do that. Yeah. Thanks, everybody, and I'm happy to talk later. Yeah.